everyone. Welcome to Learners Hub Vellore. Hope you're all doing great. This session is all about hemoglobin. Hemoglobin. It is a protein found in red blood cells that carries oxygen in our body. It carries oxygen and it gives red color to the blood. Average hemoglobin content in blood is about 14 to 18 gram per deciliter. However, it varies depending upon the age and sex of the individual and the number of RBCs. Let us see its normal range. At birth, it is 25 gram per deciliter. For male, it is 13 to 17 gram per deciliter. And for female, it is 11 to 15 gram per deciliter. This is the structure of a hemoglobin molecule. If you see in a red blood cell, there are totally 4 helical shape of polypeptide molecules. There are 4 polypeptide molecules. There are 2 alpha chains and there are 2 beta chains. Okay, and each polypeptide molecule, it comprises of iron. And there is one heme group also. Now let us see its explanation. Hemoglobin is a conjugated protein. It consists of a protein called globin and an iron-containing pigment called heme. So this is that heme. Okay. And iron is present in an unstable ferrous form. Ferrous in the sense Fe2 plus form. Heme part is called porphyrin. It is formed by 4 pyrrole rings, that is tetrapyrrole. The iron is attached to each pyrrole ring and globin molecule. You can see that it is attached to each pyrrole ring and globin molecule. Globin is made up of 4 polypeptide chains. We saw that among the 4 polypeptide chains, there are 2 alpha chains and 2 beta chains. That also we saw. So adult hemoglobin is written as HbA alpha 2 and beta 2 in the brackets. As there are 4 units of heme in one molecule of hemoglobin, there are 4 iron atoms in one molecule of hemoglobin. So each carries 4 molecules of oxygen because of the unstable ferrous state, that is Fe2+. Coming to the synthesis of hemoglobin, hemoglobin synthesis actually starts in the pro-erythroblastic stage. However, hemoglobin appears in the intermediate normoblastic stage only. The production of hemoglobin is continued until the stage of reticulocyte. The heme portion of hemoglobin is synthesized in the mitochondria. So where it is synthesized? In the mitochondria. The protein part is synthesized in the ribosomes. Protein means ribosomes and heme is synthesized in the mitochondria. Coming to the synthesis of heme is synthesized from succinyl coenzyme A and glycine in the mitochondria. Formation of globin. The polypeptide chains of globin are produced in the ribosomes. There are 4 types of polypeptide chains, that are alpha, beta, gamma, and delta chains. Each globin chain molecule is formed by the combination of 2 pairs of chains. Now coming to the formation, adult hemoglobin contains 2 alpha chains and 2 beta chains. This is for adult hemoglobin. Coming to fetal hemoglobin, it contains 2 alpha chains and 2 gamma chains. Each polypeptide chain combines with one heme molecule. So after the complete configuration, each hemoglobin molecule contains 4 polypeptide chains and 4 heme molecules. Factors necessary for the formation of hemoglobin. Various materials are essential for the formation of hemoglobin in red blood cells, such as first-class proteins and amino acids of high biological value. They are responsible for the formation of globin. Who are they? First-class proteins and amino acids. They are responsible for the formation of globin. Iron. It is responsible for the formation of heme part of hemoglobin. Copper. It is responsible for the absorption of iron from the gastrointestinal tract. Various materials are essential for the formation of hemoglobins in RBCs, such as cobalt and nickel. They are responsible for the utilization of iron, that is during hemoglobin synthesis. Vitamins. What types of vitamins? Vitamin C, riboflavin, nicotinic acid, and pyridoxine. They are responsible for hemoglobin synthesis. Coming to the destruction of hemoglobin. After the lifespan of 120 days, the lifespan of erythrocytes, we saw it is about 120 days. The red blood cells is destroyed in the reticuloendothelial system. Where is this? It is particularly in the spleen. It is the graveyard of red blood cells. And hemoglobin is released into the plasma. Soon the hemoglobin is degraded into reticular endothelial cells and they get split into globin, iron, and porphyrin. After degradation, they become three, that is globin, iron, and porphyrin. They get split up. This globin is utilized for the resynthesis of hemoglobin, and iron is stored in the body. Porphyrin, it is converted into biliverdin. From where this porphyrin came? It came from the degraded red blood cells. Porphyrin is converted into biliverdin. In human being, most of the biliverdin is converted into bilirubin. Now, there are two things, biliverdin and bilirubin. What are they? They are called as bile pigments together. Now, coming to the functions of hemoglobin, they transport oxygen from lungs to tissues. Of course, they do. They transport carbon dioxide from tissues to lungs. Yes, they will do that. Hemoglobin acts as a buffer and it plays an important role in acid-base balance. Yeah. Hemoglobin gives red color to the blood. This is very important. And hemoglobin maintains the shape of red blood cells. Hemoglobin degradation accumulates physiologically active metabolites. Hemoglobin has an important role in transfer of various drugs. Coming to the clinical conditions, high hemoglobin level causes. This is for high, that is polycythemia, lung disease, kidney tumors, hypoxia, heart disease, dehydration, smoking cigarettes, high altitude, taking drugs like erythropoietin. Now coming to the clinical conditions of low hemoglobin level causes, that is anemia, kidney failure, loss of blood, traumatic injury. Because loss of blood is because of these many, that is traumatic injury, surgery, bleeding, colon cancer, stomach ulcer, bone marrow problems, like a replacement of bone marrow, that is because of cancer, they do it. Suppression by red blood cell synthesis, that is how they get suppressed, that is by chemotherapy drug. Coming to abnormal hemoglobin structure, that is sickle, because of sickle cell anemia or thalassemia, you can tell. Abnormal hemoglobin. A hemoglobin abnormality is a variant form of hemoglobin that is often inherited and may cause a blood disorder, that is hemoglobinopathy. Hemoglobin is the iron containing protein compound which is present within the red blood cells. They carry oxygen throughout the body. It is made up of heme, so which is the iron containing portion, and globin, which are the carriers of proteins. They contain protein in it. The globin protein consists of a chain of amino acid, that is, they are the building blocks of proteins. There are several different types of globin chains. They are named as alpha, beta, delta, and gamma. Normal hemoglobin types include hemoglobin A. They make up about 95 to 98% of hemoglobin. It is mostly found in adults. It contains about 2 alpha chains and 2 beta protein chains. Coming to hemoglobin A2, that is HbA2. They make up about 2 to 3% of hemoglobin. They are found in adults. It has 2 alpha chains and 2 delta protein chains. In the previous, we saw that it has 2 alpha chains and 2 beta chains. This is for hemoglobin A. Here we are seeing it is having 2 alpha chains and 2 delta protein chains. This is for hemoglobin A2. Coming to hemoglobin F. It is fetal hemoglobin. It makes up to 1 to 2% of hemoglobin found in adults. It has 2 alpha and 2 gamma protein chains. It is the primary hemoglobin. It is produced by the fetus during pregnancy. Its production usually falls shortly after the birth and it reaches adult level within 1 to 2 years. Genetic changes, or you can call it as mutations in the globin genes, they cause alterations in the globin protein, resulting in structurally altered hemoglobin. Coming to hemoglobin S, HbS is a disease which has 2 abnormal beta chains and 2 normal alpha chains. The presence of hemoglobin S causes the red blood cell to deform and assume a sickle shape, that is like a crescent shaped moon or sickle shaped circle, you can tell. Whenever exposed to decreased amounts of oxygen, that is, it might happen when someone exercises or has infection in lungs during vigorous exercise, or if we have infection in lung, the red blood cells form this sort of sickle shape. That is when there is lack of oxygen. Not always, okay. Sickled red blood cells are rigid and can block small blood vessels, causing pain. They can cause pain. They can block these small blood vessels, impaired circulation, and decreased oxygen delivery, as well as shortened red cell survival. So these are all the changes taking place. Hemoglobin H. It is an abnormal hemoglobin. It occurs in some cases of alpha thalassemia. It is composed of 4 beta globin chains and is produced due to a severe shortage of alpha chains. Due to the shortage of alpha chains, this is happening. Although each of the beta globin chain is normal, the tetramer of 4 beta chains does not function normally. It has an increased affinity for oxygen. There is an increased area or affinity for oxygen, holding on to it instead of releasing it to the tissues and cells. Hemoglobin H is also associated with significant breakdown of red blood cells. What is it is called as? It is called as hemolysis, as it is unstable and tends to form solid structures within the red blood cells. Coming to hemoglobin C, this disease is caused by a mutation in gene that provides instruction to the body to make hemoglobin. So this is providing instruction to make hemoglobin. This mutation causes a change in shape of RBC so that oxygen isn't carried well throughout the body. This is also because of lack of oxygen. Oxygen is not carried out in body. Then we will again feel pain and many more. Coming to hemoglobin BARTs. HB BARTs develops in fetuses with alpha thalassemia. It is formed of 4 gamma protein chains when there is a shortage of alpha chains, in a manner similar to the formation of hemoglobin H. In hemoglobin H, you can see it is similar. Similar to the formation of hemoglobin H. Okay. Protein chains when there is a shortage of alpha chains. If a small amount of HB BARTs is detected, it is usually disappears shortly after the birth due to dwindling gamma chain production because of shortage of alpha chains, and after birth it will disappear because of dwindling of the gamma chain production. These children have 1 or 2 alpha gene deletions and are silent carriers. They are silent carriers, they have 1 or 2 alpha gene deletions, and they have the alpha thalassemia trait also. If a child has a large amount of HB BARTs, he or she usually has hemoglobin H disease and a 3 gene deletion. Fetuses with 4 gene deletions have hydrops fetalis. First, they said 3 gene deletion. Now they are telling fetuses with 4 gene deletions, what they have hydrops fetalis, and usually they do not survive without blood transfusion and bone marrow transplant. So this is very severe. Hemoglobin SC disease. It is because of the inheritance of 1 beta S gene and 1 beta C gene. it results in hemoglobin SC disease. One S is coming here and one C is coming here. Because in the name itself, you can see hemoglobin SC disease. These individuals have a mild hemolytic anemia and moderate enlargement of the spleen. Persons with HBSC disease may develop the same vaso-occlusive, that is blood vessel blocking complications as seen in the sickle cell anemia. Most cases are less severe. Lack of oxygen because of that, right? So hope you all enjoyed this session. Thanks for watching and stay tuned for our latest updates.